Welcome to Ramchandar's Microbiology channel, Microbiology Quiz 8 on Microbial Diversity, Eubacteria, Part 1. Question number 1. Escherichia coli, a typical gram-negative bacterium, has size of A. 0.25 to 1 micrometer, 1 to 2 micrometer. B. 0.25 to 0.5 micrometer, 2 to 5 micrometer. C. 0.5 to 1.5 micrometer, 2 to 5 micrometer. D. 1 to 2 micrometer, 2 to 4 micrometer. Answer is A. 0.25 to 1 micrometer in breadth, 1 to 2 micrometer in length. Generally, in measurement of bacteria, breadth is told first, followed by length. Answer is A. Question number 2. Gram staining distinguishes bacteria on the basis of A. The nature of their cell membrane, B. The nature of their cell wall, C. The type of nucleic acid they have, D. The presence or absence of capsule. Answer is A. The nature of their, the answer is B. The nature of their cell wall. The nature of their cell wall. Answer is B. Gram staining distinguishes bacteria on the basis of nature of their cell wall. Based on cell wall composition and cell wall structure, gram staining distinguishes bacteria into gram positive and gram negative bacteria. Question number 3. Peptidoglycan content is more in cell walls of gram positive Eubacteria, B. gram negative Eubacteria, C. gram negative Archaea, D. gram positive Archaea. Peptidoglycan content is very high in gram positive Eubacteria. Gram negative Eubacteria, peptidoglycan content is less. In Archaea, peptidoglycan is absent. So answer is A. Peptidoglycan content is very high in gram positive Eubacteria. Question number 4. The thickness of cell walls of gram positive Eubacteria ranges from A. 10 to 20 nanometer, B. 5 to 10 nanometer, C. 10 to 40 nanometer, D. 20 to 80 nanometer. Answer is D. 20 to 80 nanometers. Gram positive Eubacteria have very thick cell walls compared to gram negative Eubacteria. Question number 5. The thickness of cell walls of gram negative Eubacteria ranges from A. 10 to 20 nanometer, B. 5 to 10 nanometer, C. 2 to 5 nanometer, D. 7 to 8 nanometer. Answer is D. 7 to 8 nanometers. Cell walls of gram negative Eubacteria are very thin compared to gram positive bacteria. The thickness of gram negative eubacterial cell walls ranges from 7 to 8 nanometers only. Answer is D. Question number 6. Peptidoglycan, also known as murine, peptidoglycan content in total dry weight of cell wall of gram positive bacteria and gram negative bacteria are A. 10 to 20% in gram positive and around 5% in gram negative, B. 20 to 40% in gram positive and around 5% in gram negative, C. 30 to 50% in gram positive and around 10% in gram negative, D. 40 to 90% in gram positive and around 10% in gram negative. Peptidoglycan content is very high in gram positive cell walls compared to gram negative bacterial cell walls. Answer is D. Peptidoglycan content in gram positive cell walls ranges 40 to 90% in total cell wall material of gram positive bacteria. Whereas in gram-negative bacterial cell wall material, peptidoglycan content is only 10%. Answer is D. Question number 7. Teichoic acids are present in cell walls of A. Gram-positive Eubacteria, B. Gram-positive Archaea, C. Gram-negative Eubacteria, D. Gram-negative Archaea. Answer is gram-positive Eubacteria. Peptidoglycans are present in cell walls of gram-positive Eubacteria. Gram-positive eubacterial cell walls are exclusively made up of peptidoglycan and teichoic acids. Teichoic acids are characteristic of cell walls of gram-positive Eubacteria. They are absent in gram-negative Eubacteria and Archaea. Answer is A. Question 8. Teichoic acids are polymers of. Teichoic acids are polymers of A. Glycerol phosphate, B. Ribitol phosphate, C. Glucosyl glycerol phosphate, D. Glycerol phosphate or ribitol phosphate or glucosyl glycerol phosphate. A or B or C. Answer is D. Teichoic acids are polymers of either glycerol phosphate or ribitol phosphate or glucosyl glycerol phosphate. In some bacterial cell walls, teichoic acids are polymers of glycerol phosphate. In some other species, teichoic acids are polymers of ribitol phosphate. In some other species of bacteria, teichoic acids in cell walls are polymers of glucosyl glycerol phosphate. Answer is D. Question number 9. Teichoic acids are neutral, B. Negatively charged, C. Positively charged, D. A or B. Answer is teichoic acids are negatively charged. Teichoic acids are negatively charged because they have either glycerol phosphate, phosphate is negatively charged, or ribitol phosphate. Due to that, they are negatively charged. Question number 10. Lipids are rich in cell walls of A. Gram-positive Eubacteria, B. Gram-positive Archaea, C. Gram-negative Eubacteria, D. Gram-negative Archaea. Answer is lipids are very rich in cell walls of gram-negative Eubacteria. Gram-negative Eubacteria contain very high amount of lipids in their cell walls. Gram-positive eubacterial cell walls, lipid content is very less. Answer is C. Question number 11. The outer cell wall of gram-negative bacteria is primarily composed of an outer membrane made up of A. Proteins plus lipids, B. Phospholipids plus proteins plus lipopolysaccharides, C. Phospholipids plus proteins plus lipoproteins, D. Phospholipids plus proteins plus lipoproteins plus lipopolysaccharides. Answer is D. Outer membrane of gram-negative bacterial cell wall is made up of phospholipids, proteins, lipopolysaccharides and lipoproteins. Answer is D. Question number 12. Porins, beta barrel proteins that act as channels allowing diffusion of small water soluble molecules are found in A. Outer membranes of gram-negative eubacteria, B. Outer membrane, mycomembrane of mycobacteria, C. Outer membranes of mitochondria and chloroplastus, D. All the above. Answer is all the above. Porins, these are beta pleated protein channels that allow the diffusion of nutrients in gram-negative eubacteria. They are present in outer membranes of gram-negative eubacteria. They are also present in mycobacteria. Mycobacterial cell wall is made up of 3 layers. Outer membrane, that is also called mycomembrane, in that porins are present. Outer membranes of mitochondria and chloroplasts also contain porins for transport of nutrients. So answer is D, all the above. Question number 13. Peptidoglycan is a polymer made of sugars and amino acids that forms the cell walls of bacteria. Specifically, it is composed of alternating sugar units N-acetyl glucosamine and N-acetyl muramic acid with peptide chains cross-linking the sugar strands. In peptidoglycan, N-acetyl glucosamine linked to N-acetyl muramic acid by A. alpha 1,4 glycosidic bond, B. beta 1,4 glycosidic bond, C. alpha 1,3 glycosidic bond, D. beta 1,3 glycosidic bond. Answer is B. beta 1,4 glycosidic bond. In peptidoglycan, N-acetyl glucosamine is linked to N-acetyl muramic acid by beta 1,4 glycosidic bond. N-acetyl glucosamine, the first carbon is linked to fourth carbon of N-acetyl muramic acid. So these sugars are linked by beta 1,4 glycosidic bonds. First carbon and fourth carbon linking. First carbon of one molecule is linked to fourth carbon of other molecule. Question number 14. The outer membrane of the gram-negative cell wall is attached to the underlying peptidoglycan by means of which of the following? A. Bronze lipoprotein, B. Proteins, C. Phospholipids, D. Lipopolysaccharide. Answer is A. Bronze lipoprotein. Cell wall of gram-negative bacteria contain 2 layers, outer membrane and inner cell wall peptidoglycan. The outer membrane is linked to inner cell wall peptidoglycan by means of bronze lipoprotein. Answer is A. Question number 15. Gram-negative bacteria are more resistant to antibiotics due to the presence of A. Thin peptidoglycan wall, B. outer membrane of cell wall, C. porin proteins, D. teichoic acid. Answer is outer membrane of cell wall. Gram-negative bacteria are more resistant to antibiotics due to the presence of outer membrane of cell wall. Answer is B. Outer membrane in gram-negative bacteria do not allow antibiotics. It is generally more impermeable to transport of antibiotics into the bacterial cell. So answer is outer membrane of the cell wall. Question number 16. Which structure is found in the outer membrane of gram-negative bacteria and is responsible for the endotoxic effects of certain bacteria? A. Lipoteichoic acid, B. Lipopolysaccharide, C. Bronze lipoprotein, D. Proteins. Answer is lipopolysaccharides. Lipopolysaccharides of gram-negative bacteria which are present in the outer membrane of cell wall acts as endotoxins. Answer is LPS, lipopolysaccharide, B. Question number 17. In the peptidoglycan of gram-negative bacteria such as Escherichia coli, tetrapeptide chains are A. cross-linked by pentaglycine bridge, B. cross-linked by pentapeptide bridge, C. directly cross-linked, D. some are free, some are directly cross-linked. In the peptidoglycan of gram-negative bacteria such as Escherichia coli, tetrapeptide chains are free and some are directly cross-linked. Answer is D. Question number 18. In the peptidoglycan of gram-positive bacteria such as Staphylococcus aureus, tetrapeptide chains are A. Cross-linked by pentaglycine bridge, B. Cross-linked by pentaproline bridge, C. Directly cross-linked, D. Some are free, some are directly cross-linked. Answer is A. Tetrapeptide chains in gram-positive bacterial cell walls. Tetrapeptide chains in the peptidoglycan of gram-positive cell walls are cross-linked by pentapeptide bridge, pentaglycine bridge. Answer is they are cross-linked by pentaglycine bridge. Answer is A. Question number 19. The tetrapeptide chain in the peptidoglycan of gram-negative bacteria such as Escherichia coli typically contains A. L-alanine, D-glutamic acid, mesodiamino-pymalic acid and D-alanine. B. L-alanine, D-glutamic acid, L-lysine and D-alanine. C. L-alanine, D-glutamine, L-lysine and D-alanine. D. L-alanine, D-glutamic acid, L-lysine and D-alanine. Answer is A. Tetrapeptide chain in gram-negative tetrapeptide chain in the peptidoglycan of gram-negative bacteria contain L-alanine, D-glutamic acid, mesodiamino-pymalic acid and D-alanine. Answer is A. Question number 20. The tetrapeptide chain in the murine of gram-positive bacteria such as Staphylococcus aureus typically contains A. L-alanine, D-glutamic acid, mesodiamino-pymalic acid and D-alanine. B. L-alanine, D-glutamic acid, L-lysine and D-alanine. C. L-alanine, D-glutamine, L-lysine and D-alanine. D. L-alanine, D-glutamic acid, L-lysine and D-alanine. Answer is C. L-alanine, D-glutamine, L-lysine and D-alanine. First amino acid L-alanine. It is common in both peptidoglycan of Escherichia coli and Staphylococcus, both in gram-positive and gram-negative. The second amino acid in gram-negative bacteria it is D-glutamic acid. In gram-positive Staphylococcus aureus it is D-glutamine. The third amino acid in E. coli it is mesodiamino-pymalic acid, whereas in Staphylococcus it is L-lysine. So the answer is C. Question number 21. Percentage of lipids in dry weight of cell wall in gram-negative bacteria. A. 15 to 20%, B. 10 to 15%, C. 5 to 10%, D. 2 to 5%. Percentage of lipids in dry weight of cell wall in gram-negative bacteria is 15 to 20%. Answer is A. In gram-negative cell wall, lipid content is very high. It is 15 to 20% in total cell wall material. Question number 22. Percentage of lipids in dry weight of cell wall in gram-positive bacteria. A. 15 to 20%, B. 10 to 15%, C. 5 to 10%, D. 2 to 5%. Answer is D. 2 to 5%. In gram-positive bacteria, in the cell wall, lipid content is very less. It is 2 to 5%. Answer is D. Question number 23. The cell walls of gram-positive bacteria contain very less lipids, 2 to 5%. But mycobacteria and corynebacteria in their cell walls has high lipid content. It is A. 60% in mycobacteria, 40% in corynebacteria. B. 40% in mycobacteria, 60% in corynebacteria. C. 50% in mycobacteria, 40% in corynebacteria. D. 60% in mycobacteria, 50% in corynebacteria. Answer is A. 60% in mycobacteria, 40% in corynebacteria. In gram-positive bacteria, cell walls contain very less lipids. It is generally 2 to 5%, except mycobacteria and corynebacteria. Mycobacteria and corynebacteria Even though they are considered as gram positive, they are acid fast. Mycobacteria is acid fast bacilli. It is considered as gram positive. This bacteria contains 60% lipids in its cell wall. In total cell wall material, it has 60% lipids. Whereas Corynebacteria also has up to 40% lipids. Some species of Corynebacteria contain 40% lipids in the cell wall. Answer is A. 24, question number 24. When gram-positive bacteria are treated with lysozyme or grown in presence of penicillin in isotonic medium, these are converted to A. Protoplastus, B. Spiroplastus, C. Mycoplasmas, D. All the above. Answer is protoplast A. So when gram-positive bacteria are treated with lysozyme or grown in presence of penicillin in isotonic medium, they lose their cell wall. Lysozyme dissolves the cell wall. Penicillin inhibits peptidoglycan biosynthesis. As a result, the cells treated either with lysozyme or grown in presence of penicillin, they lose the cell wall. They become cell wall-less bacteria. These are known as protoplastus. Answer is A. Question number 25. When gram-negative bacteria are treated with lysozyme or grown in presence of penicillin in isotonic medium, these are converted to A. Protoplastus, B. Spiroplastus, C. Mycoplasmas, D. All the above. Answer is B. Spiroplastus. Gram-negative bacteria contain two-layered cell wall, bilayered cell wall, outer membrane and inner cell wall. So when treated with lysozyme or grown in presence of penicillin, only the inner cell wall is dissolved. Still, the bacteria contain some portions of the cell wall. When it is grown in presence of lysozyme or when it is grown in presence of penicillin or treated with lysozyme, it dissolves only peptidoglycan. So still, gram-negative bacteria contain lipids and lipoproteins attached to the cell membrane. So such cells which are partially lost to the cell wall are known as spiroplastus. The acid fastness of Mycobacteria and to a lesser extent acid fastness by some Corynebacteria is primarily due to presence of dash in their cell walls. A. Mycolic acids, B. Oleic acids, C. Arachidonic acids, D. Stearic acids. Answer is A. Mycolic acids. Presence of mycolic acids in Mycobacteria and Corynebacteria is responsible for their acid fastness. Acid fastness means when they are stained, that they do not lose the primary stain even after washing with HCl and alcohol. Even after washing with acid and alcohol, they do not lose the primary stain. That we call it as acid fastness. Acid fastness of Mycobacteria and certain extent Corynebacteria is due to presence of mycolic acids in their cell walls. Answer is A. Thank you.